Mm-mm-mm-mm. Hello, can you hear me? Hello, can you hear me?
Oh, hello. Yes, I can hear you.
Oh, cool. Okay. How are you today?
I'm doing well. How about you? Are you feeling better?
I am. Yeah, I'm feeling much better. Thank you for asking. Um, well, apart from the fall of the government and all like the social
Yeah.
unrest that we're about to go through over the next few years, but
Yeah.
health-wise, I do feel much better. So
Good,
was I
good.
too honest and open? Hello, can you
I'm
hear me?
glad
Does this
at
work?
least you have that. No, yeah, it's interesting times to say the least.
Yeah, so I went on this like listening course. Also I'm on my work computer, so I'm phoning in, so I can't turn on I don't have a webcam.
Oh,
Anyway, so I went on this training course, and it was about like active listening and being a intentional listener. And so now when people keep asking me how I am, I'm overly honest. Oh dear. I'm sorry. Oh
I
hey,
think that's good. Yes.
um hey, just down below, have you met Mister before? Mister, hello. Are you there? Can you meet Rachel? You two would get on really well. Um, uh, so Mister did some really great like exploratory work. I was trying to work out how best to visualize the keg pathways and those sort of like pathway reactions beyond just keg. And Mister, I found some cool like what's it called Python GitHub like repos and such. And so Mister kind of volunteered to play with them with some of the open science data from OSDR. I wonder if she's got that
Oh,
ready. That would be such a cool thing for her to share with the group.
that's
Are you
great.
there? I wonder if because yeah, there's a lot of different ways to kind of like use the transcriptomics data to kind of what's it called to kind of make predictions as to sort of biochemical changes we might see in the cells and how that is important in different tissues or developmental stages. And so that's why I'm a big fan of KEGG, because it kind of often encapsulates some of those loci and puts them into illustrations that show the developmental stage and the tissue, and thus like the pathway that was likely to be altered when we see this one little gene that's being up or down regulated. But the problem with that is then kind of weighted by the imagination of the person that made the keg illustration. So it's great because it gives us a kind of uniform perspective over certain like standardized pathways, but then it's also kind of limited because of that. It doesn't necessarily embrace the full kind of cellular biology sometimes. And so the code base that Mr. explored was an alternative way to do that, essentially, that didn't use the kind of keg pathways but had kind of cellular illustrations. Um,
Oh, that's very cool.
and it was really neat.
I would
They
love to
haven't
see that.
responded. I'd love them to show them. I've got like four different note taking things all lined up here trying to get in.
Oh no. Yeah, there's always a lot.
Oh yeah, one, two, three, four. And I'm going to reject them all. I know some people really like them, but again, this would be the good thing about having like one designated note-taking thing, which is kind of standardized and consistent. Uh, yeah. So that way, it's. But again, um, I am getting better now. Now we have this in Teams about exporting like the transcript and the video. And the last one that we had, I did put the video out. There was one really bad edit where I just had like 10 seconds of Rachel and Rocha looking very confused or handsome or whatever it was. But the second edit did get the whole video. And so now I'll be looking at getting YouTube videos put onto the Gitbook page as
Oh, cool.
an insert. So that way it's kind of all kept together. Uh, anyhow, so how are you guys today? Interesting times. Okay, I'm gonna drop. Oh, Kristen is here too. Oh, cool. I want to ask her about the APH data and the progress on uploading that from Norm Lewis. So make sure we bring that back later because that'd be a fun thing to discuss. Thank you, Rachel, for dropping in the link to the document, the running documents anyone can kind of contribute. Miss still hasn't responded despite me praising her work earlier. So maybe she's too shy to share with the group today, but we'll try and circle back to that. So looks like it's going to be quite a quiet day today. Let's hope it is the case. Because it is like just after spring break, so I'm sure people are working very hard in their classrooms. Okay, cool. What do people want to discuss today? Before I put Kristen on the spot. Hello, oh dear. Hello, Kristen. The reason I wanted to put you on the spot was, Chris, have you heard from the APH team, Norm Lewis and those guys? I'm just wondering if there is a rough timeline for, and if not, no worries.
No, it's not
Okay,
that I have forgotten. It's just that there's only so much I can
totally
do.
Okay.
There's only so much it's good that you can ask. Yeah.
Yeah. Okay, cool. No worries. Yeah. I might reach out to him and just I won't. Like, it's not my place to. But yeah, it's interesting.
And you can. I mean, I can. Sometimes maybe hearing it from someone else, but I'll. It has been a few weeks since I've checked in, so I'll send another kind of reminder today.
Cool. And I do have a reason to reach out, which isn't just pestering, kind of following up with the seedlings, because there was a stage when they were like a week old when the seedlings were thinned. So I have an excuse to follow up with them as well. So and that's just one of the things we want to do as a community, is kind of like help each other get through the process of submitting the data, uploading it. Sometimes it's difficult, and so yeah, that's kind of part of the reason for this community existing, to help each other use open science resources. Yeah, thanks, Chris. Okay, cool.
Yeah, of course. Thanks for reminding me. Honestly, there's a lot going on, so I appreciate that.
Yeah, there's a lot of moving parts, and these are weird times. I don't let me go off on that tangent. Okay, stay focused. Space biology is cool. I had a good way to bury my head in the sand. Oh, hi Dana, how are you today?
Good. Sorry, a little bit confused. I couldn't find the Teams link, but other than that, I'm good. So I'm here now, and I'm happy.
Awesome! Yay! Happy and healthy. That's the main thing. That's what we like to hear. So we're just kind of doing a round robin, because we don't really have a concrete agenda. I think the link was shared earlier, so we can just have a quick chat. I know that Ked popped his head in earlier. I just want to he wants to say anything, or I don't know.
I have a little announcement if we have time.
Yeah, please go for it.
We've been working with JSC a little bit to get some kind of archival historic Apollo plant data. A lot of it's described in the publication I just put into the chat that Annalisa and Rob Farrell did a review on. It's not of course, but it's not any omics data. It's things like pictures of a variety of different plant species that had been grown not in like lunar regolith exactly, but in kind of a growth medium, and then some lunar regolith had been like basically sprinkled atop it to see, because back in the late 60s, early 70s, there was a lot of concern that regolith was oh, sorry, someone has a hot mic. There was a lot of concern that the regolith would, bring kind of infestation to Earth. And so this was all conducted, as you all know, in kind of a quarantine facility. And they were very cautious in their treatment, as is described in that publication. But there's still some pretty interesting, growth data, biomass data, some kind of elemental analysis, and then some photos. And we're trying to get that organized in a way that we can get it into our repository and hopefully make it useful, especially as we look towards these future Artemis missions and think about how to kind of build on the work that was done in that time.
I think that is a great idea. Yeah, it's particularly important when we have these papers that are stuck behind a paywall. Yeah, just kind of popped open, and I know that GeneLab initially was set up with the goal or prerogative of focusing on the omics data, but as we have come to appreciate from many of the different groups here in discussions, that it's really important to anchor that omics data and physiological data. So I'm pleased that thanks to the Ames Life Science Archives integration, that we now have that kind of extra prerogative or reasoning to kind of delve into some of these papers and try and see if there's any kind of important data that could be archived. And so I think that is a great paper that you found there, and would appreciate it being any of its results being integrated into the Open Science Archive. To the other plant analysis working group subject matter experts that are here today, that's you guys. Do you concur? Would this be a great use of Chris's time? I see a smile and a nod from Rachel. That's great.
Yeah, this is really cool. I'm excited to go through and read this.
Yeah, the pictures are really cool. I mean, there's 3,000 images and then a bunch of, I mean, you know, the type of data you would probably guess, but the taxonomic breadth of the research is really interesting. There's like algae and then pine seedlings, and it's, you know, peas and tomatoes, and it's
radish.
Yes, and radish, some old friends, and then some kind of unexpected ones, which is I think will be really cool.
Yeah, and particularly useful / important for the new focus group, the lunar regolith subgroup. Rachel Walker,
yes.
Do you guys want to have a wrap and just say about how use this idea from Chris, or this paper Chris, as a bridge to promote that, just to so that's where that's at, how you progress is made over.
Yeah, absolutely. So we're starting to meet next Monday, April 14th. Let me check the schedule so I don't say things wrong. At 10 a.m. Central Time. So yeah, it'll be exciting. So I'm planning the first meeting to basically there were like a very good feedback and a lot of people that are interested in the topic and topics to cover. So there were like very good suggestions for topics. So I'm basically going to put them on the table and just start discussions. So we're going to start meeting bi-weekly, and you know, I anticipate it will be a fun one like always.
Yeah, that's cool. Anything you want to add to that, Rachel? Any extra stuff? Yeah, I just didn't know that it's a really popular topic, right? How many you've had, like a lot of people reach out through the OSDR forum, right?
Yeah, we have like over 30 people reaching out, and actually there are some people that are not just within the plant AWG. We have people from other, like mostly as well human AWG as well, you know, microbes, like quite a few of them as well. So it'll be interesting to see what kind of merging we do with all of those topics.
Yeah, I mean that's a book. Have you considered writing a book?
I was about to say
Hey,
that
hold
there's
on,
so
hold
many
on.
different
Like
sections.
Richard, you know, that's something that is on top of my head. The problem is like I don't. You definitely don't have time either, you know. But like, I think, you know, if we were to put it together, you know, maybe in a year we could maybe get it done. Like write a couple of interesting chapters each. The problem is like, you know, I have no experience writing a book, so I'll be
I
of little to no help.
just with like 30 like lunar regolith researchers in a room, there are so many potential stories and narratives, like trying to herd them into one paper. Well, you're a brave man. You're a brave man, person. A brave team, Rachel. You're brave people. And again, just so I'm thinking, like sometimes people's ideas. Sometimes you have that many great scientists in a room. There's more than one idea, one storyboard, and so
Yeah.
just being prepared. I don't know.
Yeah. Well, you know, in my current work, I'm learning to deal with different people with different backgrounds and very diverse projects, you know. So it's been fun that way. There's
Yeah,
like
I think
quite a
you
bit
did
of crazy, quite a bit of craziness going on around work, as Rachel may know. A lot of PR stuff, but
I think you did a great job putting together lots of different ideas on the plant-human interaction paper, that earlier one. So hopefully that review continues to move forwards, and we'll see how that turns out.
so yeah, we got reviews back last night, so I guess early morning time in England, and both of them say one of them is I'm convinced it's AI saying, yeah, there's grammar mistakes here and there without pointing at lanes, but I said, yeah, overall it's fine. And the other one is saying, yeah, it's good to go. Just clean up a couple of the figures, which is true, and you're good to go. I add a tiny bit more of intro for a certain topic. So Chris already told me, it's like, yeah, it's like, can you resubmit this? So I guess they gave me 14 days to resubmit. So I should meet the deadline for sure. But yeah. So that seems to be almost out of the door. So that's good.
That's
Yay!
great. I saw that email, so I was really curious as to what the comments were. So that's great. I'm glad to hear.
Yeah, they are there. It's like very, to be honest, very lame comments. Like there's really nothing. Like it's,
I guess that's
you know,
good for us.
yeah,
Because it's gentle.
but at the same time, it's like, one of them for sure, like, I don't know if you read the paper, probably there was a deadline for next day, like you have to submit the reviews and, it's okay, much better. Like when I last submitted to Nature Microgravity, the other paper, I submitted in June. It's still under review. It's like we got some comments back. It's crazy. My boss doesn't know what to do anymore with it. It's like, what do we do? Do we pull it out? Do we send it somewhere else? And they say, no, it's almost, reviews are over. The reviews were fine. And then we're just waiting for the decision. So it's crazy.
Um, It really does depend on the reviewers you get. It can make a huge difference, can't it? It's really,
Mhm.
or maybe it's the story too. There's angles. I don't know.
And then I guess
But,
Richard, I'm going to promote you and me a tiny bit. Like and Jennifer Claudio is leading a Meet the Authors Day on April 24th with Richard, John Keys, and myself. So it will be hopefully fun. I don't know yet what kind of questions they're going to ask us. So um, it'll be a good time for sure.
Yeah, so I just can't say no to Jen. She's such a wonderful educator and person, and yeah, always an honor to have the chance to discuss some of the work that we've done, to help promote the Open Science Archive and how it's enabled some of this research. And yes, everybody's welcome, particularly those that want to bring softball questions. We love softballs. It's a thing. Okay, cool. So what's next on on agenda? Oh, who's just turned up? I just had some new people join the virtual room. Is anyone new here? Oh, I would like to hey Suji, how's it going?
All right, thank you.
So we're about to steer the conversation away from the regolith, because that's we've had that conversation. So with Sushi turning up, I always want to turn the conversation back to roots. I'm terrible, aren't I do like roots. They are important. But that's not essential. So yeah, we should look back to the agenda and see if anyone has any key concepts. Or Abhinav is typing, so do feel free to speak up, mate. It's good to see you came back. We didn't scare you off last week. Cool, more people are coming. Okay, great. So yeah, we have many different stakeholders and concepts within space biology. So there's many different tangents the conversation can go off on. We've done the SAM thing, which is promoting some of the success we've had through focus groups, and the latest focus group that's coming up is the regular one. But again, just kind of promoting that people want to use this community to create focus groups in areas of their interest. They're welcome to. Yeah, actually, I think my June couldn't come this week, but she's coming back. She's just and so she'll be trying to get some people to do a cross-species narrative with her. So maybe we could, if people are interested in doing some kind of a cross-species story where we kind of merge humans, plant, and Drosophila and mice into one big matrix and look at what the core genes that change, or orthological groups of genes that change across all different species. Do reach out, because June needs a bit of a help, and I think it's going to be a really cool story. I see Rachel nodding. I do love your enthusiasm, and you are great at this. So keep it up. Yeah, I think you'll really like June as well. So I'll tap. That is also the great thing about having your cameras on people, because sometimes your facial expressions give your soul away or your thoughts away. You don't even need to speak out. Okay, yes. So that was just an example of one of our stakeholders who isn't here, who has a specialized interest in combining different species data and then analyzing that, and trying to find. So that's just one way. Again, any one of us can kind of find a kind of subdomain within biology, and then kind of drill down using space biology data to kind of find new insights from the data that's stored within the Open Science Archive. Again, kind of harking back to our kind of core guiding principle and there. Okay, cool. So we've got the agenda here. I was looking back through some of the previous things of this discussed previously. The pseudo time series stories is still on the freezer, because we're still waiting for the Apex 3-2 data, RNA-seq data to go through the system and be published. So that's the favorite story I want to go after.
So I thought, Richard, you were going to present about your experience at the Biosphere.
I can do, yeah. I was trying to check to see if there's anything else first before I'm charging off onto the Biosphere tangent, because it is so much fun. But I am concerned it's going to feel a bit like me showing photos of my holiday, but it was really informative, and actually the journey of going through it taught me some things about future space bases in theory, assuming that they're not just going to be designed to be metal boxes with no ecological components, just like small metal boxes for humans to squat in for a few days as they get irradiated in distant planets or moons. So I like to think of space bases being things that are designed to be like lived in with different communities, like intentionally cohabiting, and not just like national communities, but also like different species. And that's one of the great things that we saw at the Biosphere. Okay, so the first thing I'm going to do is drop this link into the chat. Okay, 1 second. So what is Biosphere? Well, here's a beta product that was made from it. So this is Simoc. I've promoted this previously. I'll come back to this moment. You can just kind of sign in as a guest if you kind of press forwards. So this is a computer model that was made using the data from the Biosphere 2. So Biosphere 2 is a giant greenhouse in the Arizona desert, which was made back in the, I'm going to get some slides up too in a second or two, but hopefully you guys kind of Googling around, because there's so much information on it. Whatever I put into a slideshow isn't going to do it justice. Just an FYI. So yes, it's a giant greenhouse in the middle of the Arizona desert, which was built
Are you sharing your screen, Richard? I'm not seeing it, but I'm on my phone.
No, I'm not. Basically, because I'm how to put it, Google. I'm looking for like the Google photo photo archive. And yeah, it's just taking me a little bit longer than I thought. But so ultimately, where are we going with this? Biosphere. That's what I was looking for. Biosphere 2, 2025. Okay, cool. Excellent, fun times, and I'm gonna share this in a second, and then that will help us see where we're going. Okay, cool. So a few weeks ago, or a few weeks before this, and there's no share button. So a few weeks before going to Biosphere, one of my friends was share screen or window or tab. Let's share the whole screen, the entire screen. Whoa. Okay, cool. I just click on that share button. So entire screen. Is that sharing? I don't think it is yet.
No, not yet.
Okay, but maybe I'll just do a window. Yeah, but just do a window, that's better. Okay, cool. So a couple of weeks before going to Biosphere 2, I saw this photo from a friend of mine on LinkedIn. So this is in the SAM. So the SAM is the prototype that was made for Biosphere 2. So it's like it's an analog research environment, which is completely airtight. So it's hard to make completely airtight things. And you need oxygen.
Richard,
and CO2.
The screen is not showing. Oh, there it is.
Cool. So and what you can see here is an experiment going on inside that from the US Air Force, where they've got algae on some shaking incubators, and some they're tilted to give a lunar or Martian gravity simulation. And so basically you can see there's an interest in checking the efficiency of algal cultures in sealed environments, because this is one of the potential ways that we could generate oxygen in situ and reduce the amount of CO2. So this was just something that was happening just before we got there. So why did we go there? What is this? That was just the day before we left. So I set up the RPM. Okay. So we set up some experiments in the lab. We went off to Arizona. So we knew that we were going to this facility that was designed for doing investigations to what it would be like to build a habitat on the moon or Mars. And so I'm not going to go through the historical stories of Biosphere 2 and go see the either the Biodome movie or there's some movies on YouTube about it. But on the way there's this agricultural research institute out in the desert. And we went to go visit, took the students, yay, Purdue ag students looking at flowers, gotta love it. We spoke to the local professors who started teaching us about how to grow hydroponically in a dense scale. So this was fun for the team to kind of see this really awesome way of growing tomatoes. These are kind of indeterminate ones, so they wrap the stems around the hydroponics, so they're constantly fruiting all the time. But it wasn't just kind of modern agriculture that we were interested in. The reason we went to this ag station is they have indoor ag as well. And they've been like fine-tuning the light environments. There's many people that have been looking at how to make the light optimum for crop growth in vertical agriculture. But they'd also put a lot of time into the movement of gas. And so they've been modeling about how actually when you have these kind of high density growth systems, the movement of air becomes very limiting quite quickly. Oh, and it's important for cacti too. It's beautiful cacti. I ended up hugging some cacti while I was there. But the reason that's
was that
important
Murat Kassira, by the way?
Pardon?
Was that Murat Kassira that Murat Kassira from University of Arizona?
Yeah.
Okay.
University of Arizona, and then they have this like lunar HAB simulation. So they've some literature on this essentially with and this was a prototype that was made before the Lunar Palace. So the Lunar Palace is the Chinese version of this, which I went to go see. And it was inspired by this. So this was made in order to basically demonstrate that you could create these growth environments that fit inside a rocket. So hence the cylindrical nature. They can be expanded out or closed in, and then they can be used to grow plants. So we got to go see it. It was very exciting. Yay! But essentially, it's just like hydroponic systems in a tube. But going in there was kind of really inspirational for the students. I do recommend going to visit these things, and you can make them yourself. But essentially, the goal of this, so there's these two growth cylinders, and on the far end there's a third one that they've attached, and they're now building like a human HAB facility in. So they're basically now redoing what was done in the Chinese Lunar Palace. But these were the guys that made the design to do that. It's just it was defunded by NASA back in the day. I'm not going to go. There's a new paper that has been reviewed that will help explain the story as to why, rather than just having this kind of half-built demo version in Arizona, we don't have a fully functional version on a NASA base. But I'm going to get off that soapbox. That's something from the past. I'm going to focus on the future. It's great that this design was replicated in China and shown to work. And it was awesome visiting it. For me as a space biologist, to go to visit this was kind of like on my bucket list. Yay. Again, so Ray Wheeler and some of the pioneers in the field behind this, and somewhere on the Open Science Archive there is a PDF that was described all the details as to the initial design for this. Danik, I'm trying to remember the name of the program that was in the paper that we were reviewing. Anyway, so after visiting this great example of what a kind of lunar base is kind of going to consider to be like, inside a metal cylinder with loads of kind of like technology holding it together, right? We then went to see what could be a very different vision of what extraterrestrial bases could look like. So this is a view. So over here, you can see some of these like greenhouses that are surrounding Biosphere2 over here on the right. Here's the moon in the distance. And so people have dreamed that maybe one day we'll be able to build on a biosphere on the moon. Okay, cool. But what are the things we have to consider when building a new biosphere on Earth? As you can see, Mars is very happy. Marshall Borders very happy. And Anjana next to here, the undergraduate who arranged the whole thing, being inspired by this beautiful site. And so the team went along, and we got to go inside. One of the things we learned is this guy here has been modeling it. Kai is an absolute legend. If you get the chance to visit or contact him, do. He is an absolute wealth of knowledge, and it was a real honor to get to wander around with him and learn from him. He inspired each one of these students in each of their own different ways. And if you look at that SAM software I shared earlier, he is kind of the person who helped build that. So this biosphere behind us has got loads of sensors throughout it. So they know like the CO2 levels, the oxygen levels throughout it, because some biospherians tried to live in this thing for 2 years. That was the goal. Unfortunately, the goal during that 2 years was to not open it to the outside world, to make it fully like sealed, to see if humans could live in a sealed habitat for 2 years. And there's a movie about it on YouTube, which is free. So I go watch it, Spaceship Earth. And well, they didn't survive. They didn't work through the full 2 years, but they learned some valuable lessons along the way. So I wouldn't call it a failure. Okay, cool. But what did we learn? Building a greenhouse in the desert is difficult. It turns out there's like massive temperature swings from day to night. So if you're trying to build this structure, each one of these pieces of glass has been put in there. You have a pressure differential from day to night, which is basically enough to pop each of them out. Why would you go for such an audacious structure? Well, one of the engineers was Buckminster Fuller himself, that famous engineer that designed those kind of buckyballs. So you can read, as soon as he mentioned that, you can see that in the design here. The only other way to get an arch structure of that kind of size and shape is using the arches, which were prevalent in Moorish architecture, but they went for the Buckminster style instead. So I mentioned that the engineering challenge associated with keeping these windows in place during the day-night transitions, and they did that by making this giant lung. So this is a giant dome, which inside it has a mechanism that moves up and down, a diaphragm like a lung. Actually going inside it is really fun because there's this massive whoosh of air in each direction because it's a pressurized chamber. And I'm sorry that my photo is so terrible, but what you see in the center is like a pool because this is like a condensating area for the chamber as a whole. And so water accumulates here in the center and gets pumped back into the main greenhouse. And this diaphragm above us goes up and down. You can kind of see the metal legs there. So when the Biosphereans were living in this chamber, this was moving up and down, preventing the windows from popping, and was helping them recirculate the air that they were breathing in the different biomes and helping recirculate the whole process. There's a secret tunnel that connects that to the main building, and I felt like a bit of an explorer going through it. It was surprisingly fun. I'm such a big kid. And as you can see, each of the different sections are pressurized. So a bit like going from one section of a submarine to another. So again, trying to get us to feel what it would be like to be on a greenhouse on the moon or Mars. It's kind of some of the shots we might get if we were lucky enough to go visit one of those future extraterrestrial destinations, assuming humanity can produce them. I love this moment behind the scenes, all the pipes, the concrete, and the trees just kind of, and the sunlight shining through. It was awesome. Incredibly noisy. There's a lot of machinery that goes into making this ecosystem function in the desert because of those massive temperature swings. And depending on where you are on the moon, you will likely to have these massive temperature swings. Unless you go into a crater where it's just cold. Okay, this is the ocean ecosystem. You've probably seen this before. I recommend you write proposals to go scuba dive in there and do some kind of science because it would be so much fun. People do that. We can see above it these massive fiberglass metal framework that they've had to build above it. It's actually hideous. The guy was really sad when he saw it for the first time because it kind of ruined the aesthetics of it. But it's so they can mount massive lights over here. The coral reef didn't thrive when they tried to do this experiment previously. So now they're building in these massive new lighting system. So the coral reef is going to be actively energized as they try to identify coral reef varieties that are going to be able to withstand the heating that the sea is undergoing. So right now there's people that are sampling coral reef from around the world because there's a massive coral reef die as a result of elevated CO2 in the atmosphere, and the greenhouse gas effects are causing the atmosphere to warm as well. So those two features are causing acidification of the top 10 meters of the ocean, which is enough to disrupt most of the coral reef on the planet. So there's a group of scientists that go around sampling the coral reef and trying to identify the varieties that are most likely to survive and be able to grow and thrive in these future warmer seas. And then trying to bring them indoors to scale up production, so we have stock to move out to the future degraded coral reefs. So again, just so what this shows is that although Biosphere2 may not have worked perfectly to demonstrate a successful way to build a biogenerative life support system that didn't need internal atmospheric inputs, it did create a wonderful microcosm to allow us to explore some of the potential consequences of climate change moving forward. I was obsessed by the algae and the mosses that were growing on the edge of the aquatic ecosystem. I thought it was awesome. But the most inspirational one has definitely had to be the jungle ecosystem. It was just so cool. They were producing. The trees had got to a state where they were producing so much biomass that they had to go up and climb this framework to go to the top to cut down the trees. So again, another kind of unexpected job opportunity for those of you that like rock climbing and have a head for heights. There's a need for tree surgeons in these types of habitats. It was fun to visit. What we also learned is now they're trying to establish a canopy underneath, which is productive for things like coffee and bananas, and not just for the ecosystem services that it provides, aka the filtering of the air. This is another view of that metal rig above the ocean ecosystem that was built. Hopefully from here you can now see why I was kind of referring to the shock and sadness that our tour had, because it's not exactly pretty, is it? And this just goes to show what happens when you have lawyers involved in building design. Essentially, because people go under here for experiments like scuba diving and such, this thing has to be incredibly stable in order so the insurance agency got involved and made this massive framework just because they wanted to hang some LEDs above the water. Okay, so there's a lesson there. Don't let the lawyers get involved whenever possible. Oh, excuse me. Sorry, just having a laugh there. But yes, apparently this framework is very important for allowing, because they need to have some good UV lights and to help grow these new corals. Study in progress. So this was one of the funny things. We ended up standing on the right here, and looking down while discussing. And next to us, you can see these canna lilies. Here they are next to the study. And it turns out we were standing next to what would have been the Biosphere2's toilet. So just over here to the right is an area where they would have been going to relieve themselves and just using the natural ecosystem and plants like cannas to filter the water. And actually one of the Biosphere2's that left this has actually spent the rest of their life going around the world helping to create grey water filtration systems like this, because they saw that as a great way to help reduce some of our impact on the environment. Pineapple. Who doesn't get excited when they see a pineapple? Aren't they cute when they grow like that? Oh gosh. Sorry, just tangent. So yeah, it was fun exploring the different biomes. The savanna ecosystem wasn't really grown with the goal of feeding humans. It was kind of just there for aesthetics. So you could have put olives trees in there and it'd been a lot more fun. A lot more productive for humans, I should say. Oh, but the flowers. It was so weird being in the desert, being in an area where there was not a lot of life, and then going into the greenhouse, seeing this orange plant flowering was just, it just had so much more impact on me psychologically. But ultimately, from the space biology perspective, we're here to kind of look at the systems, the engineering systems, and how the biology can fit into that. And so this diagram kind of shows how it was originally kind of planned. And it's kind of fun when I look at this, I see an electrical circuit, but instead of having electricity moving from section to section, what you see is actually biomass that's moving. And so the goal of this is to essentially, use natural ecosystem services to make sure that all the things that the astronaut or the explorers in the greenhouses need is provided. So that's water, oxygen, removal of CO2. This is actually a photo from when they're building it. Sorry, it's really blurry, but the point of it was to show just the amount of concrete they were putting in. And so this system was so well designed. In theory, it should have worked, but what they didn't account for was that the concrete in the building was going to be taking in oxygen. And so as it was drying it, and so essentially there wasn't enough oxygen to continue making so the oxygen in the habitat started to decline. And so basically there was a point when they had to bring in fresh air. And this is the sort of thing that you don't really want to do on the moon. And some people like Ray Wheeler have done some mathematics to essentially predict the cost of this, because all habitats leak. And so this is one of the reasons that it's postulated that future lunar bases are going to have a half atmospheric pressure. They're not going to be at 1 bar like we're used to. This is in part so astronauts can put on spacesuits and go outside and walk around and hop around and bring rocks back and do all the cool things all the science that we want them to do on the lunar surface. But that creates a secondary challenge, because you can see here, if we're going to have those explorers in a habitat on the lunar base, we're going to need to feed them. I'm sorry this photo is blurry, but you can see that when these people first made Biosphere2, they had a cropping system at its heart, like an orchard producing vegetables and fruits and grains. And essentially, when we think about these lunar bases, this is the niche that I think a lot of us here believe the crops are going to fill, producing the nutrition for the astronauts and also producing some of the oxygen for them to breathe, and while removing the CO2 from the atmosphere to stop it getting to a toxic level. But how many of us in this room know the consequences of growing these plants at half a bar? And if it knows us to say, okay, yeah, I've got a rough idea about what's going to happen if I grow a, say, Arabidopsis plant at half a bar pressure. What about then when you put animals and humans into the equation and you start to vary the CO2 and O2 content? How well will your yield models hold up in that type of environment? I don't know. It's an interesting area, and it's really hard to research because you kind of need these sealed environments in order to do that. And because the Biosphere2 is no longer sealed, it's now open to the public so people can go through. It's no longer useful as a research bed for these types of atmospheric flux experiments that are required in order to design truly sustainable biogenetic life support systems. Oh, what an inspirational site, Biosphere2. Like the vision these people had all those years ago, like 30 years ago, and see this, like the people that built it, they didn't actually have experience in building. They were artists, and so they acted, and they were actors. So they acted like business people in order to make it all happen. One of them acted like a designer to help make it happen. It's brilliant. They then built a boat to go around the world to collect the different species. And they ultimately put into the starter of this ecosystem. A really an inspirational group of characters that built this place. And the view from the tower that they built is so cool. To go around. So, hello, how's it going? I don't know who that was, but nice to hear from you. So this was the room that they got to stay in. So I think when we think of people going to analog environments, maybe not this luxurious, right? So it definitely helps to have a wealthy landowner and sponsor on board when designing and building hubs. Oh, so this tower in the center, this is where I was taking those pictures from earlier. That was that was their library. And halfway through the experiment, so at the beginning, they go up there when they finished doing their chores, they finished cooking the food and growing the plants, then they got up there to read. And that was fine to begin with, but it got to a point where the oxygen was being used up, like I described earlier, when they literally couldn't climb the steps anymore because they were so weak. And that was one of the things that led to them needing to bring in more air and kind of giving up on the primary goal. So there we go. That's the summary picture that shows like that journey that I went on. Thank you for allowing me to share my holiday snaps, and hopefully there were some good lessons learned. Okay, but that's just kind of like the holiday moment. There's a research section to it, and that was the next area that we went to. See, hopefully you can see this door on the left. So this is the door to SAM. So the SAM, I alluded to it briefly earlier, is much smaller, and it was the prototype that was made to test all of the engineering that was used to build the main Biosphere2. So the SAM is just over here, just outside the room, and all you can see here is a new Martian, what's it called? Martian playground. I mean, Martian film set. I mean, a Martian work yard. That's the term that they're building. They haven't built in the regoliths for the ground yet, but you can see they've got they've had a load of set design people from Hollywood come visit and build the walls. All the walls are also designed based on actual NASA photos from the rovers that have visited. So these are true geological features that have been photographed. They also have a rig that allows analog explorers to kind of climb into it. And there's some weights that will counterbalance you so you can walk along and hop like an astronaut. It's really fun. So again, they're trying to create a facility here for people to visit because it's cool. So this is just next to that Martian yard. This is the SAM. We're just going to the door here. So this is all fully airtight and sealed. Went inside. So right at the beginning, I showed you that video of the algae. There's people from the US Air Force, right, doing algal experiments. This is that same room a couple of weeks later. So essentially, anyone can propose to do experiments there. But I'm not encouraging everyone to do that, am I? Okay, cool. In here, this is the main workroom. They have some hydroponic systems on the sides. Each of these are custom built. And it was really great. So they tell you some things they learned just as they built them. It's anyone who's built hydroponics know the third one that you build is the best because the first two you learn things while building them. And so that was really inspirational for some of the students who are currently designing hydroponic systems like these guys as part of the Purdue search team. So these are student astronauts that wanted to come on this trip. Actually, this is one of those student engineers who just graduated who's actually working at Biosphere2. So this is actually a great picture showing that, actually, when you get a degree, your job it can actually lead to a job at places like this. This guy was awesome. This was marshaled. What happens when you take a biologist into the desert? They find plants growing in trees, and isn't it great? So funny. Yeah, that was mistletoe. So yeah, this is the final thing. So this is that Mars hat, that Mars thing, and so hopefully the students, there'll be some pictures of them going on that we did. Okay, maybe I didn't take pictures of that. Obviously, I had to do my breakdancing. I'm sorry, can't help myself. But this is the area where they're going to be testing all the new CO2 scrubbing facilities. So this gentleman here just done a really good demonstration talk about how he's building a CO2 scrubbing system that's going to be plugged onto the side. So this is the SAM facility in the background. So this is where we were earlier with the greenhouses. Next to it, they've got a fabrication facility and a shipping container. And then they have a second shipping container, which is your habitat where people can stay. On the right here, we've got the lung. So this is a mini lung because this has the same issue as the big Biosphere 2 in the sense that because of the day-night temperature profile changes, that if it didn't have a lung next to it, it would the windows would probably pop. So the more of the story is, there is this really cool facility right here in the desert. Actually, sorry about my breakdancing. I'm terrible, I know. I'll get to a picture of this SAM where it's not being just for fun. So yeah, this SAM facility has the opportunity is an opportunity for everyone to go visit and propose to do experiments inside this thing. It can get fully sealed up and can do all kinds of cool atmospheric flux things. Anyway, that's the gist of that. I haven't really got much more to pitch. Any questions? I can stop sharing any second. I'm just wondering if there's another.
Hey, Richard.
Hopefully you can. Hey, go for it. How's it going?
Yeah, I just you mentioned the half bar, and so just in text there, I thought I'd look up the highest altitude settlement at 5,100 meters. It's close to half a bar. So go to Peru and grow stuff. See what happens up there.
Well, I'm glad you said that. Because someone from NASA has done that. Not Brian. It was the other one. Okay, I'll stop this share now. But you can see all these students are really inspired by it. And you're right, it's this atmospheric component, like this high altitude. So there's also, so I've been looking at some plants that have evolved in high altitudes for that exact reason. Okay, how do I stop this share now? Oh, it's so much fun taking students to analog environments. Well, this is the dude telling about the CO2 scrubber he was building, because it's really important. This was the picture. There we go. The students, so this is now, the weight here is designed to give a lunar G-force feel. So you can kind of walk along and jump and bounce like you're on the moon. So that's one of the other fun things for people to do when they go to visit the Biosphere 2. Hey, Sigurd, you got your hand up. Sorry, how's it going?
Yeah, I was just curious about visiting. So what is access to the public like?
It's really easy for people, for the public to go. Essentially, you can just turn up any time and pay a small fee at the door. Try and resist the gift shop, and then you can go straight in. What's interesting is the University of Arizona also has a campus there. It was built by, I think, the University of New York, something like that. But essentially, it was a field site for other for another college to go to the desert. And so there is actually a really nice accommodation area next to it, which is why, again, it's a really great place to visit, because you can rent a chalet there, as it were, in a nice mud abode building, and be right next to it. And so that way, it's a really good place for people who want to visit and use this, either visit the Biosphere 2 or use the SAM as a research platform. They're also building a robotic arena, so a lunar garden, so where robotic teams can go and practice different types of robotic maneuvers. So the gentleman Kai that runs this facility is an entrepreneur. He's brilliant, a really friendly, clever, soulful person. So again, if any of this has seemed at any point I'm pitching to you that this is a great opportunity for you or your students or friends to do analog research experiments, then if you go to the CIMOC Space Simulator webpage, you can find Kai's information through there. It's a bit cryptic because he's really good on computers, so he knows not to just put his email address on there. So I'll drop that link in there. Anyway, cool. Ted, thanks for the link about the 0.5 bar. So there's a couple of varieties of Arabidopsis that have evolved at those high areas. So until I've tortured them, I won't be raving about them. I want to know what their response is before describing how these high altitude communities are really important. Peru. Who was that from Peru? Bruce. That was Bruce, wasn't it? Yeah. So you mentioned about how in Peru is at a high altitude, and so this half bar, which is what we're predicting for a lunar or Martian settlement. So yeah, in Peru, Bruce went to go visit because there are communities that are farming up at the Atacama Desert level. I do have his slideshow on that, but that's a slightly different tangent. So there is an opportunity to do that. So you can try and create half a bar and try and do that sort of work, or you can go to Peru, where there's you can go down to sea level and then you can drive up to that high altitude in the same day. So there is again a real opportunity associated with that geographical location, which we should probably follow up on. Anyway, I digress. Whoa. I'll stop. So was that fun? It was a little bit different. I normally like to just dive into genes and plant physiology, but it turns out those things are really infected by the environment. And Suchi has a hand up. How's it going?
Hi. Yeah, thanks. No, it was really interesting. I just had a quick question because I don't think my comments are ending up in the chat for some reason today. I just wondered if you thought that hydroponics, I've asked this question before, so sorry if it's repetitive, but do you think hydroponics is like a realistic scenario then for plants in a, potentially in a future lunar base going forward?
Well, that's the $1 million question.
Okay, fair enough.
Which way of doing them, but no, yes, I think it is. And rather than just saying that's my opinion, I'd allude to other pioneers in the field like Ralph Ritchie, who's kind of have stated this multiple times and produced data to kind of support it. When we think about the future lunar base, for me the best analogy is when the U.S. Air Force first built hydroponics on scale on route to Guam when they were sending soldiers out to the Far East, because they massively scaled up hydroponics on one of the volcanic islands and were able to feed people en route. And in doing so, they did so with hydroponics. So if there was an argument to do it with soil, that would have been the situation when soil would have been applied, right? And yeah, if we had Ray Wheeler in the room, which we don't at the moment, he would again also allude to some of his papers and work that's helped kind of support the yield gains that you get through hydro. Hopefully, but again, this is opinion and this is science. There are ways to create data that they potentially could contradict that answer, and I'm always willing to change my opinion based on data. But now currently that's my perspective, and see, I'm a real scientist. I'm willing to accept doubt in my equation perspective. Oh dear. But yeah, so if you have a contradictory opinion, feel free to create said data, and I would love to collaborate and or read said paper.
Oh no, not at all. No. I'm just curious because as I'm interested in roots, so I'm thinking ahead of what system to grow plants in. Basically, if we can simulate lunar gravity on Earth, because the kind of data, modeling in the loosest terms that we're working on with James is looking at root architecture and lunar gravity, right, or predicting what root architecture could be in lunar gravity. But all of that is based on obviously images that we have from the APEX missions, which are agar, like plants, Arabidopsis grown on ATS or media plates, right? So it's just thinking just wondering if there are aspects of that we can incorporate. So absolutely, I absolutely don't have any data to the contrary, and I don't expect to have it anytime soon. But thank you for that. That was very informative.
I mean, there are a lot of different root system architecture phenotypes, and yeah, there's bound to be some that we would attribute just to gravity or lunar gravity changes. But then a lot of the ones like, I know, primary roots, lateral roots, ratios, those types of things, there's so many other environmental factors that kind of feed into that. that's where normal terrestrial agriculture data kind of feeds in, and thus hydroponics tend to win on those types of growth metrics. But anyhow, I know that Hamid is here, so we do have a hydroponic specialist here, and so again, I
was just
defer to people with more data. Good for me.
So Richie, there's a lot of ways to affect root architecture, and it doesn't necessarily have to do just with gravity. So there's techniques to force plants to grow certain types of roots just based off of what kind of system you have and what you think optimizes their ability to take up roots or even stabilize portions of the plant. So this conversation could go for like 3 hours if you wanted, but you can feel free to reach out to me. I'll put an email in the text, and if you have questions, I can probably answer most of them.
That would be amazing. Yeah. So one of the things that we're trying to, hopefully I'll be able to present at a future meeting. Is one of the things we're trying to do is dissect like the effects of the different stressors, and see what potential effect individually they might have on root architecture. So of course, just from the Apex missions, gravity is one, but then so looking at geomagnetic fields, CO2 and light, like things like that. So it's quite interesting because absolutely, like we're just getting an idea of these different things and how they might influence root architecture. So absolutely. No, I'd love to chat further. So that would be amazing. Thank you.
Yeah. Even like plant density will change how roots interact with each other. I mean, I don't know if you've seen them before, but like they'll just make a network with each other. They'll share nutrients with each other. They'll share information with each other, I guess, through markers. But anyways, I'll put the email in the text.
That'd be amazing. I guess root exudates would be something to consider as well in hydroponic solutions, right? Because you absolutely, density. That's a really good point. Absolutely. Thank you.
I wonder what sort of by changing the root seedling density, I wonder what changes we would see in root exudates. And the other little comment there I'd have on that is thinking about what would be the goal of said crops in said lunar ecosystem, and how would that affect the cropping systems that are adopted. Obviously, I'm going to lean into microgreens. You've heard me rave about it, but you would imagine a certain portion of the ecosystem would be for that, and that would have a different cropping system, and thus a different effect on the roots and how you'd water them and such. But then you could imagine, hopefully after looking at Biosphere2 and SAM, maybe there would be other green crops of some sort that would be grown to provide other ecosystem services, and things like food that they might produce might be like a secondary benefit as opposed to the other ecosystem services that they provide. Don't know. Just thinking out loud, kind of trying to reflect on Biosphere2 and the ecological lessons we have there, because we could save some money, because shipping atmosphere to the moon is going to be incredibly expensive, and that's why in situ recycling is really important. And some of the hardware that we use to do that on the ISS isn't really sustainable for lunar missions, apparently, according to some philosophers in the area. Obviously, the industry is moving fast, and you guys might know technologies that change that paradigm. But for now, that's why we're discussing this area. Okay, so this has been fun. We've been here for 55 minutes. Our time is nearly up. Does anyone have any other kind of questions or tangents or comments or things they'd like to share? I don't know.
I'm just quickly curious, how high did the CO2 get when the Biosphereans were living there?
That is a good question, and it was really high. I'm literally just Googling it, CO2 levels.
Okay.
But it got to a level where it affected their their behavior. So they started getting agitated and fighty with each other. They got and so again, think of it as being isolated on the moon or Mars with a small community, small group of people, and then not knowing that your atmospheric composition is changing, and not knowing that can affect your behavior. It's one of those things that we don't often have in our equations. When you look at all of the different things that we discuss, the the behavioral effect of elevated CO2 and then low oxygen isn't something that comes up much. And that was one of the major lessons from the Biosphere2. But the exact amount of CO2 it went up to, I'm not 100% sure. But again, one of the reasons this is kind of poignant is because of the preliminary data that we got from ExoLab 11. Isn't it, Ted? Pivoting around. But like, it was awesome. And one of the things that I was really surprised when Ted shared some of these data was how high the CO2 got in that small box. It was like up to 10,000 ppm. And when I mentioned this to some friends at Kennedy, they weren't surprised. In fact, apparently there are some sensors that show things that are that high or more or different. Obviously, it's kind of stochastic because it's atmospheric, but that's why I'm super excited that GeneLab shared the rodent data through their environmental data app. So I recommend checking that out. That's one of the few snapshots we have into the atmospheric environment of the ISS. Granted, it does have the mice running around in there, so they are releasing some of the CO2. But it would be interesting to see on some other sensors elsewhere. Hopefully one day again. I think that's why Chris. That's what earlier while I was asking in part about the APH, but just chasing the CO2. I see. Yeah,
we are working on getting APEX, all of the environmental data we have for plants formatted for the EPA. I have no timeline for that, but we're working on it.
Yeah, brilliant. Appreciate that. And I mean, when that's there, again, we can perhaps get the crew here together to make GeneLab to help do meta-analysis and review all those. Yeah, because it's going to be really interesting when you look at the experiments that are exposed to the ISS atmosphere and those that are exposed to those that are locked into small boxes. And yes, because as Ted was saying, like the U.S. Navy has allowed up to 7,000 ppm for people on their submarines. Oh man, like. And they're saying that thing about the Biosphereans kind of getting angry and agitated because of the change in their atmosphere. Oh dear, we don't want Navy officers people getting angry and agitated. I do hope that they don't go up to that 7K ppm limit that often. Hopefully they probably keep it a bit lower. Yeah. So I guess like you can see by the way this conversation is turned, one of the main lessons from the Biosphere2 is that I have come to appreciate atmospheric composition a lot more than I did previously, because when you work with a conviron growth chamber, all the atmospheric circulation is kind of just built in naturally, so you take it for granted. Yeah, and maybe we shouldn't. Okay, cool. So that's the end of the run. We've got to the hour. Is anyone have any other comments, questions, or points I'd like to talk about, or do we want to call it a day and do something mindful or spend a bit of extra time eating or digesting during our lunch break?
I just want to say thank you so much for sharing the pictures. That's very exciting. Of course, I've have followed some of the Biosphere2 work, and just online and stuff and on social media. So it was very cool to see actual pictures.
Cool, yeah, and again, I'm sure I didn't the academic in me thinks I should have made an academic presentation with beginning, a middle, and end, and a clear message and a clear goal. But I wanted to put my heart on my sleeve and just show the experience I had and what I learned from it. And hopefully, maybe that means some of you guys might think it's easy enough for you guys to go to, because anyone can go visit the Biosphere2, and it's just brilliant. Brilliant. I'm sure some of the students that went on that trip with us, it was like a pivotal moment, because afterwards they're talking about how they changed their career goals and all kinds of things. So truly inspirational. Hence, being excited to have the chance to share that story without getting too academic about the whole thing. Okie dokie. Oh, Prul's just arrived, but we can. Hey Prul, how's it going? I was just kind of winding up, to be honest. But yeah, does anyone have any extra things, any final comments or statements or things to add? Or shall we call it a day and take back this extra half hour? Hopping off.
Yeah, we're good. I think next time I want to see a video of you dancing.
Yeah, we'll do so. We'll do. I did get full spins on that pick and everything, but yeah, got to enjoy yourself, right? Always be yourself no matter where you are. Okay, great. Well, thanks everyone, and yeah, we'll be back in a couple of weeks to yeah. If anyone would like to sign up, they can. Mustair, maybe that'll be a good time to try and bring up that analysis that you did previously and try and get some people to help collaborate with that, because I was really excited by that code base that you developed and those results that you showed. So perhaps there's a way yeah, perhaps you and Rachel can reach out, and food for thought. Okie dokie. Well, have a good day and bye for now. Thank you.
Thank you.